All right, welcome back to part four as we dig into the diagnostic clinical lab about antibiotics. And it's an important aspect of really what we're trying to do is to find out some good treatment paths that we can take for the patients, and we do this in the diagnostic lab. But we also use it to help identify bacteria as well. So don't let me underemphasize that we can use antibiotics for lots of things. So antibiotics, of course, are a good component of public health. We do have concerns. It is a public health concern. Antibiotic resistance is a huge issue. We have seen resistance now to every major pathogen to I would say every different antibiotic we have. And partly this has happened as a result of sort of just the approval process in the veterinary side, where it's USDA, which is not as rigorous as the FDA, the Food and Drug Administration, is for human use of the antibiotics. So a lot of times before a human antibiotic actually even gets into the marketplace, the antibiotic resistance is seen from the animal side because it's already being used for growth promoters or treatments or various things, and it's proven to be a real problem. And so we also, of course, still use antibiotics, and we use different types of treatment techniques to try to stop an infection using antibiotics. And we also have to, of course, be aware of toxicity and allergic reactions and things for each. So it encompasses a lot of information. Conventional identification of bacteria consists of performing, of course, the Gram stain, and we also use antibiotic susceptibility testing. And a lot of times we can help identify a certain organism based on resistance in bacteria due to maybe a chromosome based and one on the main chromosome, the genes that confer that particular type of resistance, versus many bacteria have plasmids, which are extracellular chromosomal inheritance. In other words, a small subset of genes that reside on a circular piece of DNA separate from the chromosome circular piece of DNA, and it's much smaller. And usually the main thing that they confer is resistance characteristics like tetracycline or trimethoprim or whatever. But they also can carry certain adherence mechanism genes for pathogenicity and toxins and various other things associated with these plasmids that confer certain characteristics to the bacteria to give it some characteristics that lead to disease. And we, of course, we don't like that, but that's what happens. We also understand that the bacteria love to share their genes, not the Levi genes, but the regular genes. And in doing so, they confer certain characteristics, like E. coli can have Shigella-like characteristics because of transferring a plasmid to Shigella. They're not proud. They will share genes amongst different genera across the board and utilize genes. And it just comes to the concept that bacteria are lean, mean, fighting machines, and they will use whatever they can in order to grow in a niche. If that niche happens to be you, so be it. And we call that disease. Bacteria just call it, if they could talk, just surviving. So it's just a mindset that I've kind of developed over the years. So antibiotic susceptibility standards it's determined by the FDA, the Food and Drug Administration, and the Clinical and Laboratory Standards Institute. So it's not a willy-nilly type of thing. There are groups and committees and the like that monitor and come up with standards. So we say that a particular organism is resistant, let's say Shigella flexneri is resistant to ampicillin if it's over a particular size of resistance pattern as we see around a disc, and I'll show and talk about this. But these standards can tell you, well, it's sort of in between, or it's resistant, or it's sensitive. Just because you see a ring around it doesn't necessarily mean that it's resistant. It still may be sensitive based on what these panels had determined. There's usually a set for each different type of species and genera of the bacteria in terms of their performance on a certain or given antibiotic. So I just wanted to point that out. You may see a sheet that talks about this sort of thing, and it does a determination of antibiotic susceptibility, which is a quantitative data analysis. And so you can determine if it's resistant or sensitive based on this. And this is a good way. It's not guesswork, it's not willy-nilly, and it offers consistency, which is absolutely required in the healthcare industry. Antimicrobial was derived from Greek words anti, of course, against, micros, which is little, and bios, life, which makes sense. And a lot of the medical field has Latin and Greek terms derived in it. That's the systematic learning. I did place a chart, if you recall, a vocabulary listing of various words that are from Latin and Greek. And again, it's a way to kind of refer to these things so when you learn the words that they are systematically part of a language of Latin and sometimes Greek. So antibiotics do not include antimicrobial substances that are synthetic, like sulfonamides and quinolones, or semi-synthetic, which is methicillin and amoxicillin, or those that come from plants, quercetin and alkaloids, things that appear like turpentine or turpentoid, and or animals with lysozymes or proteins that are so. Antimicrobial is something that microorganisms produced against another microorganism. This is sort of the key centerpiece of what a probiotic is. That a probiotic will produce substances that keep other microorganisms from growing in their space. And what you want to do is to grow good bacteria, of course, producing these antimicrobials to keep out the bad ones. And that's a whole field I hold close, near and dear, that probiotic concept. But it is a semantic thing, antimicrobial versus, in contrast, the term antimicrobials include all agents that act against all types of microorganisms, bacteria, viruses, and that sort of thing. So anyhow, it's just a subtle point. So antibiotic is refers to substances produced by microorganisms that act against another. So that is antibiotic. Antimicrobials is much broader sense. Classification according to spectrum of activity, and you'll hear this a lot. First thing we want to do is gram stain in a diagnostic lab, and that gives the physician he or she a tool to say, okay, we're going to start with a broad spectrum antibiotic based on the fact that we know what the gram stain is of what we think the pathogen is. And so they can start at least a treatment, and then once more information is determined, then we can switch to a narrow spectrum antibacterial that is useful against a particular class of microorganisms that we know are causing the problem. So it's important to know and distinguish between broad spectrum and narrow spectrum because it's a very common thing in treatment of patients. Now, antibiotics, because of the different mechanisms by how they affect the bacteria and how the bacteria actually respond to it, we can have bactericidal drugs, and those that kill target organisms. In other words, cidal is death. Examples of these are aminoglycosides, cephalosporins, penicillins, quinolones. Bacteriostatic, it inhibit or delay the bacterial growth or replication, such as tetracyclines, sulfonamides, and macro. And the reason we use these, of course, even if they're static, is reduces the growth of the organism so that the immune system now can handle it and clear it. And so we're aware of these sorts of things and we take advantage. Now, there are times when you don't want to cure someone too fast. A gram-negative infection, if it's it's in the blood and it's systemic, you want to cure the patient on a slower type of basis, because what happens is that the patient, you could be successful and kill the organism that's causing the disease, but if you kill the organism too fast, then it becomes toxic. It's a gram-negative shock, and the patient could actually die from it. So it's all part of the strategy. I just wanted you to know that we have these tools to be able to treat and use a systematic way of having a patient for treatment properly. So the microbiologist takes in a very important role in identifying the tools that we can use, and so that's just important. So we know that since Alexander Fleming had a contaminant on his plate, and he had enough confidence and he was good, that he really allowed folks to realize that, yeah, even a scientist of my standing, Alexander Fleming, can have a contaminant on a plate. But he noticed that the contaminant caused other microorganisms on that plate not to grow. And that was a key observation. And I have to admit, that observation saved millions of lives in battlefields and in hospitals. Penicillin became, at that time, mass produced, and it was just an amazing set of events that occurred. And I know, just in personal thing, that my mother was sensitive to penicillin. She's one of those, and it came out that because of that, she never used. They wouldn't use it, and she came up with a urinary tract infection that was very severe, and she was the organism was resistant to most of the things. And the microbiologist said, "Well, it is sensitive to penicillin." The elder physician, he was up in the years, and he says, "Look, they've cleaned up the prep from the old days. The penicillin used to have a whole bunch of junk in there. Now they artificially synthesize it. It's pure. We're going to go ahead and risk it. If she is resistant, shows immune response to it, we can deal with it. But so even if she's sensitive, we can deal with it." But it turns out she wasn't allergic to it anymore, because they had cleaned it up, and it saved her life. And so knowing these sorts of things, it's all part of the physician working in concert with your clinical microbiologists and professionals to work out these sorts of things. And it's really, really important that you have that understanding. So when you read charts and various things, you know what's going on behind the scenes. And that's important, because that affects how the patient is going to be handled under your care. So antibiotic resistance is one of the biggest threats to global health. If you don't believe me, you can go on CDC and Google it. You'll find all sorts of things. In fact, I've placed a paper, and I'm going to come to this in a minute. That would be great if you had a chance to review it. That's an extra credit opportunity. You can just sort of summarize what you pulled from it. Just write it in a single page and email it to me at acvinyl@waketech.edu, and I'll give you extra credit for it. So antibiotic resistance occurs naturally. They've gone to locations that are pristine. There aren't any humans there, on islands and various things, and they found resistant bacteria in the ocean and the surrounding ponds and things, which shocked me. I said, "That's amazing," but it's a global phenomenon. A growing number of infections such as pneumonia, tuberculosis, gonorrhea, salmonellosis are becoming harder to treat, because we just don't have, obviously we're creating new antibiotics, but the same sort of philosophy is happening, is that bacteria will engineer their way around it genetically and become resistant. So antibiotic resistance leads to longer hospital stays, because you can't treat it, and higher medical costs and increased mortality. So it's a very serious problem, and you just need to be aware. So I have a paper that's in the directory of the lab area, antibiotic resistance crisis. And there's two parts, but the only one I posted is part one. You can find part two if you want, if you're interested. But this goes over some of the key features of why we're seeing this resistance crisis. And it's well documented, the CDC. It's a good read, and it gives you a nice history. I love papers that do a sort of a literature review, and it gives you sort of the timeline of key events, and this just even maps it out. It's a great paper to have. I love these sorts of things. But what I've identified, and we haven't gone over that much, but you can see there's plates that have these little discs, and bacteria have been streaked onto the plate, and then allows the bacteria to grow in the presence of this disc. And you can see this big zone of inhibition that grows around, that forms around the disc of this antibiotic. So what's happening, it's being diluted ever so much, some constant rate. I don't know exactly. I'm sure they've mathematically calculated it, but it gets to the point where the threshold where it is cidal or static, such that the bacteria won't grow around that. It's that zone of inhibition. And so we measure this distance, and we can calculate how much we can give of this antibiotic, and all that. It's systematic. The plate's incubated for a certain amount of time, and the concentration of the antibiotic in the organism, if you recall, I just talked about committees and various things that monitor and create standards, and that's why it's important. But the reason I brought up this slide is if you see these little colonies growing, and you can see the majority of the bacteria are resistant, and that's why you see the clearing. But there are some that are spontaneously resistant that show up. Now, these are mutations that occur, and this is where the resistance comes from. It is a subpopulation that, for whatever reason, have genetics that have caused, they have some mutations that now renders them resistant to this particular antibiotic, some better than others. And as you can see, that this particular antibiotic seems to be with this organism tends to be a little bit more resistant, or spontaneous resistance is showing up higher. And if we see this frequency, there is differences in various antibiotics. It comes down to genetics, evolution. A lot of the microevolutions and things that you may have studied in biology come to play with bacteria. Really kind of show this in a very quick time frame, the life's of evolution, and then these now become predominant. If this antibiotic's present, these are now able to grow in those environments, and it is part of that resistance crisis that I'm referring to, and this paper is referring to, and it's quite an issue. But this is the origins of it. It comes down to the genetics. And so you have a few resistant bacteria in the population, which the antibiotic will prevent the yellow ones from growing, but because the spontaneously mutated ones now can grow, they've now become the predominant population. And that's why normally when you give an antibiotic, you tell the patient to take all of the antibiotics so you kill all of the microorganisms, or the majority of them, so the immune system can clear it. And we don't see that, but now we're starting to see even you give that patient the antibiotic, that the patient starts to not get well. In other words, they're continuing the illness. This is what's going on. You have an organism now that's resistant, and it's predominating in the tissues. And if you were to sample again, which usually happens, it'll be resistant to whatever that treatment antibiotic. And it's just something that you need to be aware of. So mutations provide that resistance that happens. So the mode of action, I have a summary chart here that kind of shows these. Am I going to ask you this on a lab practical? No, but I just want you to be aware of, in a generic type of sense, that there are various different ways and these antibiotics do their job. We can inhibit cell wall synthesis, or the cell membrane function. In other words, they prevent them from moving food in or whatever in and out. We can actually make the microorganism constipated by clogging or stopping a function, and it can't export and that sort of thing. or we can inhibit their protein synthesis, which shuts them down, and that disrupts their normal multiplication. And there's certain antibiotics that do these sorts of things. Inhibitors of nucleic acid ones, uh, the rifampin, the the Burrs Wellcome had trimethoprim. They stop nucleic acid. Metabolic processes, a lot of these can shut those down, and so they have different ways of doing this, and some of these have certain resistance characteristics. These they find other ways around the metabolic process, and so these tend to be more resistant prone than others, and it all depends. But this is a nice little summary of mode of action. It's just, I like to make stickers in my notebooks for summaries. It cuts to the chase, although I try to remember both of these. Now, if you go into a hospital, you will see doctors using flip cards to refresh their memory on certain antibiotics and dosage and resistance patterns and all those things. They have to keep up on those things, and it doesn't hurt to kind of follow along. And so you can see some of the, like the chloramphenicals and tetracyclines tend to inhibit protein synthesis for various things. And it's just how they do this, working with the ribosome level where they can't, it prevents it from binding to the messenger RNA or whatever the technique. But it's just to be aware of the mode of actions. There I have listed here 5 different ones, the kind of broad settings, and we have some new ones that are on this list, the RNA synthesis inhibitors, and we're getting new ones all the time. So I've already kind of let the cat out of the bag in terms of the antimicrobial susceptibility test, and we use this disc diffusion or dilution method, and the putting a it's basically using a hole punch, creating on Whatman blotting paper, and then soaking that paper with a particular antibiotic concentration, and then you place those on a disc. Now these are commercially obtained now, so you don't do that, and you place them on the disc, and each of them has written on them what the antibiotic is and the concentration, and then you inoculate the plate. So you can see this one that I'm pointing at right now is not, it has been inoculated but hadn't been grown at 37 degrees overnight or 18 hours or whatever. And you want to know exactly how long you incubate these. We keep logs on these things, and then you go and measure the the pattern. So we see this zone of inhibition around these, and then you measure the distance across. Now, given the type of media, so we can use the Mueller-Hinton media. So we try to standardize on the media and the bacteria that we're putting on there. We know about it, and then the antibiotics that we're testing them for, we record that. And then the measurement tells us, if you recall the standards chart, we can say, yes, in fact, when we measure this, it's beyond a certain distance, and we can call this one resistant to that particular antibiotic. Or even though it's a small band of resistance, it's I mean, sensitivity to it, this is a huge band zone of inhibition of sensitivity. You can see the resistance is much higher in this particular antibiotic for this organism, and if you do the measurement, it would show you that it is sensitive, or I mean it's resistant to it. This is sensitive to the antibiotic, where this still may be resistant even though you see a zone of inhibition. It's whatever the standards have been, the concentration's so high that we don't deliver these type of antibiotics at that concentration. It's whatever the standards are that we match. So for us, it's just a matter of doing a measurement, and then you plug it into an equation that tells you, yeah, you want to give penicillin BID or twice a day based on that. So it's systematic, and we're trying to limit the guesswork. One of the things I use this slide is you can see little resistance patterns showing up, and this one has a lot. This one has a few. This one has a few. And again, it could be the age of the plate and the like, but this is now a new consideration. A lot of microbiologists will say that there are some resistance patterns showing, small single colonies are resistant, and there's a lot of them, then it may be we probably or choose not to use that antibiotic because we see the potential for that resistance. So anyhow, I wanted to bring that to your attention, and now you'll understand. That's why I wanted to go over that. So the standardized inoculum for a culture is you use, that's why we always do single colony isolates. So we choose one and we spread it amongst the plate. You can use a loop and aseptically go across and just streak it over and over again by going back and forth with your loop and rotate the disc 90 degrees and keep going, and it gives you a lawn of bacteria, and it gives you sort of suspension. You can also use a hockey stick and pipette a suspension of the bacteria on there. It's however your lab or the lab goes about it. We use a particular growth medium for standardization if it's involving a patient. It's the Mueller-Hinton auger, and again, we have to have a basis by which we standardize, and so we use the particular media, the incubation time, the spreading, and the disc, and then we use those, and then we measure these after that prescribed time, and that way we have some sort of consistency. So we measure the zone of inhibition, we read the panel, in other words what's the criteria for resistance and sensitivity, and then you interpret the results. And that's one aspect of what a clinical microbiologist will do, and it's really straightforward. It's not hard to do, but it's very, very important, and getting these results out is key. Now we can get a suspension of bacteria, and we can do a pour plate and get colonies to grow, or we can pipette organisms on the top and spread it with a hockey stick aseptically, and then you get colonies in confluent. Is what we want to do with a Mueller-Hinton type of technique. I just wanted you to be aware of these terms. You can use pour plates and have suspension of microorganisms in the media, or you spread plate. For antibiotic testing, it's either using a hockey stick or the spread plate method, or using a inoculation loop and just going back and forth over the surface. This gives you a nice even lawn, and it's kind of preferred, but I just wanted to point that out. To use the aseptic technique in doing this sort of technique is you place the bacteria, get the hockey stick in alcohol, flame the hockey stick, and then you spread the bacteria holding the lid. Now you obviously need to wear gloves and all that, but I don't know how to draw this nicely, so I borrowed this from someone somewhere, and it illustrates. It's really a very simple technique. You do this near a flame, and when you sterilize the hockey stick, you can spread them, and keeping that environment as sterile as possible. Obviously, the longer you leave the lid open, the higher probability of getting a contaminant and that sort of thing. So to identify the number of bacteria on a plate, there's a whole series of doing a serial dilution. We're not going to go over that. I do normally go into this just a little bit. You can get this more detail in Bio 275. I don't spend a whole lot of time on it, but what you want to do is to have a concentration enough that the TNC is fine, or too numerous to count is okay to use it, and spread that for antimicrobial testing, or looking at your screening for antibiotics. If you do want to identify the number of microorganisms, which is usually what you want to do, is just doing a very thorough job, then you continue on doing dilutions until you can count the numbers, and so you know what the original was. But a lot of times in a clinical setting, we just use cultures that are grown overnight in liquid, and then plate that. Can be done. So once you spread plate, then you get the antibiotics. Now this is applied usually with a way of delivering the disc by pushing down on a top button, and the discs are released, and it leaves a nice pattern, so they're equidistant amongst, so it's nice to get to that area so you can see the areas of inhibition, and those can be measured, and you can look at, we have two here. I'm not sure, I don't have a chart, but it looks like those are totally resistant, and these are susceptible or sensitive. to these antibiotics. So these would be of consideration to use clinically, but we want to use the best one. Usually the ones with the larger zones, or might be after doing your systematic measurements and the like, which ones can be offered. Now you usually just report that the measurement and in the standard report, and then the physician will plug in, or the data will be plugged in, and scripts can be written from this as to how much you would give, so it's systematically done. Excuse me. In the treatment and control of infectious diseases is caused by a pathogen, we use this testing, of course, to figure out which antibiotic we can use to go against them. And there's certain things that we know. Proteus species, if I can identify the organism fast enough, usually resistance to particular types of antibiotics, so we set those aside. So if we know the organism before we know its resistance pattern, we do have some information that we could move forward with if the life is in danger. If not, then we usually wait for the antibiotic. Now, I say it is the call of the physician. He or she makes that call. I just, the technician just provides the data in which to make an informed decision. So you have to weigh the toxicity and all sorts of things of the antibiotic, which is the doctor's call. So the other name for this is a disc diffusion susceptibility. It's the same thing. Don't let the nomenclature get you. There's a zone of inhibition, antimicrobial growth, and then you use a ruler to measure the diameter all the way across. And that's it. It's really straightforward. And then you use your chart to determine if it's resistant or sensitive. And this is sort of a review slide, but it's nice to say it and say it again so that I feel like you don't, I haven't lost you. And the Kirby Bauer technique is sort of the overall technique. When you say the Kirby Bauer, it is a standardized technique. So if I were to tell you that we're going to use the Kirby Bauer technique, that implies that we're using Mueller-Hinton. We incubate for 16 to 18 hours at 35 degrees, and then you measure them, and depending on the amount of CO2 or whatever, and then there's your plates, and then you can interpret those based on the Kirby Bauer technique, and then you have your list to determine which of these to use clinically. So penicillin would not be a good choice here, or erythromycin or tetracycline perhaps might be useful in a technique. And again, based on that measurement, then it would you plug it into an equation and it tells you, okay, we'll use tetracycline BID a certain concentration. In other words, you give tablets of 30 micrograms per ml twice a day or whatever. It's not guesswork. You don't stick your finger in the air and to figure out what's going on. So you look at the resistance of that particular microorganisms. There are strips that have the concentration that's delineated in terms of the concentration as it's being delivered, and it's a little bit more accurate. This is a diffusion, and they've calculated roughly how much, but this kind of nails it down. You can look right on the strip itself, and it affords a little bit more information, and it was a little bit more accurate. But typically, either way, it's whatever the standard operating procedure is for a particular hospital, and then you measure the entire diameter all the way across, usually in millimeters, and so that gives you that value. And then you can use your chart in the standards chart if it's resistant, sensitive, or intermediate. And again, there's no guesswork. You just do the measurement and you just follow the instructions, and that's what you report. And then of course then the doctor will make a call based on that. So the antibiotic interpretation is either sensitive, intermediate, or resistant, and that's micrograms per ml, and that's usually the minimal inhibitory concentration means in terms of the amount. And so there's the standards. We've already talked about that, and here's the full chart and this is for E. coli. And if you look at if it's sensitive or resistant, we see based on we saw it's sensitive or resistant based on whatever we saw in the measurements, and we can go from there and read them. So I have another paper. It talks about approach to drug resistance. This is really cool. It shows you in real time bacteria become resistant as they grow. It's really cool video. If you get a chance, it's just for your curiosity if you want to look at that. Um, Now, I did mention early on, and I just wanted to point this out. I'm going to come back to the cinematic approach here in a minute, but I just wanted to point out that there are antibiotics that help us identify a particular organism, like Streptococcus pyogenes. If it is not Streptococcus a different strain, it will most likely be sensitive, or I mean resistant to it, where we don't see the sensitivity. So bacitration sensitivity is something that we can again use in a court of law, so to speak. And when you're identifying it, it's just more preponderance of the evidence. So strep pyogenes is usually sensitive to bacitration, or toxo A. It's synonymous terms. And if it is sensitive, then you have just one more thing to identify Streptococcus pyogenes. Pyogenes is a bad boy. You can see that it is a beta producer on a blood agar plate. Now, I frown upon using these antibiotics markers on blood augers because sometimes the blood can interfere with that. I usually use a standard Mueller-Hinton plate for that, but that's just a detail. But either way, to save time or in class, a lot of times I just throw it on the blood agar plate. So that cinematic thing that I wanted you to see is it actually shows you the growth of the bacteria on the video. So on the Harvard. As you can see, it's growing into the media. So I just wanted you to see this so you can spend your time to look at this. It's in the slide set. But the reason why we wanted to look at this is in real time. I thought it was really cool. You can see the bacteria growing based on what I've already discussed in previous videos. Slides here that shows you how the spontaneous mutations occur around that disc, so you see them, and this is what happens once they start or are there. They can grow in that environment where the other ones that are sensitive can't, and so that's how we get the preponderance of resistance. And again, to do the technique, we use the spread plate method to get that to go. Now we can do using a similar technique while we're on this. We actually would, let me go back, we would use the pour plate method. I just wanted to show you what we'd use this method for, and that's to quantify certain bacteriophages. These are viruses that affect bacteria, and they produce something called plaques. In other words, the bacteria are infected with the viruses, they grow, and so many of the viruses are produced inside that it lyses the cell, they explode. And we see that area of explosion, I guess you could say, based on that. And so, uh, and so the bacteriophages have these plaques that form as a result, and it's just a nice way to determine if you have a bacteriophage or the plaques. So a lot of times these are useful because the reason I threw this into the slide set is that there is some use of bacteriophages as a way, instead of using an antibiotic, you can actually use a bacteriophage virus to kill the bacteria, and there are commercial products where you can give a patient a bacteriophage and they will do that. Now they also, bacteria have figured it out. They have a resistance pattern to the bacteriophage, And we utilize that system. Oops, I didn't mean to advance yet. Uh, and we call that system Crispr. It is a basic immune response that a bacteria cell will use, and we utilize that mechanism in CRISPR technique, but that's for another day. But the bacteriophage we can use as a treatment in lieu of an antibiotic treatment. It's being researched now a lot more. I just wanted to make a reference to that, and we can use the grid pattern to figure out how many plaques there are. Instead of counting each one, we know roughly how many is 1, 2, 3, 4, 5 by 1, 2, 3, 4, 5. So 5 times 5 is 25. If you count 3 in here, so 3 times 25 is 75. So you can see quickly there's about 75 plaques here, and without having to go through all that. So that's why we use a grid pattern to enumerate all that. So that's just a tip from your Uncle Ed here on how to do that. So anyhow, that is part 4, and we'll cover really quick next time, sort of going into the biochemical testing of bacteria. Really simple, it's a much shorter section, and I hope you followed me on part 4. All of these things build up for you to identify your clinical unknown that you'll be faced with during a lab practical. Okay, see you next time.